Welcome to Lesson 5 of Module 6 Genetic Change. In this video, we're going to be focusing on cloning. When we talk about cloning, we're going to be looking at whole organism cloning and gene cloning and see how effective these are and whether they can, in fact, change our populations. So cloning is the production of an exact copy, and we can do this either with one gene or the whole organism. And a process we're going to look at in this video is polymerase chain reaction, and further on I'll refer to this as PCR, and this is a form of DNA cloning. So gene cloning is where we select a gene, remove it from its source DNA, and then insert it into another organism to make identical copies of that gene. Kind of like the process we have done in our depth study this term. So gene cloning has to undertake this process. So we cut out the section of the DNA from the source organism and we use restriction enzymes. The gene is pasted into vector DNA or a plasmid, and this is known as ligation. The plasmid containing the gene is then introduced into a host cell by a process of transformation, and then the host cell can now make copies of the vector DNA when it makes copies of its own DNA. PCR is also used to amplify a particular DNA sequence, and it goes through multiple cycles of this denaturing, annealing, and then extension. And it goes through a process of thermal cycling. So we see the temperature change, and this helps the DNA strand to denature. We do have to use complementary primers, and these help to duplicate the required section of DNA, not the whole section. The alternative here is whole organism cloning. It's what we saw with Dolly the sheep way back in 1996, and it was where she was cloned from mature cells taken from the udder of a 6-year-old sheep. And each time a mammal is cloned, the process involves 3 animals, one that donates the nucleus, one that donates the egg, and then the one that plays the role of the surrogate mother. And we see here the process that was used in Dolly, and this is a process that is used when whole organisms are cloned. But this happens much less regularly than whole gene cloning does. Here we see a picture. So we saw a donor cell taken from a sheep, and we use the nucleus. We then see an egg cell taken from another female adult sheep. The nucleus of the egg cell is removed, and we do in fact put in the nucleus from the donor. To allow these to allow the egg and the nucleus to join, we have to provide an electric shock, which allows these cells to fuse. This cell then begins dividing normally to produce an embryo, and then we place this embryo into our third sheep, which is our donor sheep, and then it grows to produce the clone lamb, which is an exact copy of this donor sheep up here, as this was the cell that was being copied from the beginning. In whole organism cloning, we can also talk about artificial embryo twinning, and this is just after fertilization, the embryo can split into 2 cells before they in fact become specialized. Splitting is done in the laboratory, and then these are then implanted into surrogates to develop, and these embryos are genetically identical. We also see this in plant propagation, where we cut an existing plant and regrow it to form a new plant. This results in the plants being genetically identical. And we can also do this at a smaller level with micropropagation, where we see small portions of a plant being grown until they are ready to be transplanted. When we do clone organisms and genes, we have a few different issues that we come across. So identical twins are natural clones, so this process of cloning is not a new process. However, we do have concerns for animal welfare when we clone animals for food production. There are lots of moral and religious concerns if we do in fact move to the process of cloning humans. When scientists are undertaking the process of cloning, there's a religious argument that humans are in fact acting as God. We do not currently know the health risks for cloned animals, so we haven't progressed that far yet, so we don't know exactly what would happen to these organisms if we clone them. And then it is an expensive process, so therefore it does limit access to a certain part of the population. Yeah, the genetics of a cloned population results in a reduction in genetic diversity because we see identical combinations of genes within the population, and therefore these genotypes increase in frequency. This does then cause problems if there's a sudden environmental change, as this population will be more vulnerable to these changes. That does conclude lesson 5. Make sure you tune in for lesson 6.